One of the ways to get to know the amino acids is to categorize their chemical properties, and that's what the data in this chart is all about. On the top, you see the different amino acids, and the chemical properties of interest are listed along the left-hand side. Now, I definitely don't expect you to memorize the information in this chart, but to really understand and familiarize yourself with the amino acids, it is important for you to go through this line by line and look at the different properties of the amino acids. We've talked about whether they side chains are aliphatic or aromatic. We can see whether they have cyclic connectivity in the side chain or whether they're non-cyclic or what we'll call acyclic. The variation in terms of size is arbitrary, but you can see which ones are small, which ones are categorized as medium size, and which ones are large. Hydrophobic and hydrophilic is very important. Hydrophilic is another word for polar, and hydrophobic is a word that means that it's trying to avoid being in contact with water. Remember that proteins are found in water, and so some of the amino acid side chains are going to do everything they can to avoid being in contact with water. Those are the hydrophobic side chains, and those tend to be aliphatic and some of the aromatic ones as well. The side chains are either going to be neutral or they'll be charged. If they're charged, they could be negative or positive. The negative tend to come from those side chains that bear acidic groups that lose a proton and become negatively charged. The positively charged ones come from basic side chain amino acids that pick up a proton and become positively charged. W stands for weak. The weak acids are the cysteine side chain, the thiol group. It has a pKa of about 8. The phenolic group of tyrosine has a pKa of about 10. The next category is whether you tend to find the amino acids on the surface or whether you tend to find them buried in the interior of a globular protein. Those that tend to be buried don't want to be in contact with water. They tend to be the hydrophobic ones. The hydrophilic or polar ones tend to be on the surface. They're the ones that want to engage with the solvent water. The next row simply is nucleophilicity, whether the side chain bears a nucleophilic side chain like the aspartate groups of the or the carboxylate groups of aspartate or glutamate or the cysteine side chain and so on. Now, one of the things we might ask first off in terms of how these amino acids are incorporated into proteins is what's the typical number of amino acids that you'd find in proteins? And the answer to that question is about 320. And how do we know that? Well, here's the distribution of a sample of 2,912 different proteins. And we simply asked, what's the chain length for each of those different proteins, those 2,912 different proteins? And someone went to the trouble of actually sequencing and determining their length and then showing this histogram where the number of different proteins as a function of the sequence length is shown here. Typically then we have an average protein of about 320 amino acids, but you can see the distribution is somewhat more complex than that. It's somewhat skewed. It has a long tail, and there's some proteins that have as many as 1,000 different amino acids in them. If you take the same set and ask what's the typical composition of the 20 amino acids, what percentage of alanine do I have, what percentage of aspartate do I have, of glycine, and so on and so forth. And it turns out that there's actually a fairly complex distribution of the amino acids, and that's what this bar graph shows. Now there's 20 different amino acids, and so if they were uniformly distributed, where I had an equal composition of each of the different amino acids, I would expect to have 1/20th of each of the different amino acids, or in other words, I would expect to see that there would be 5% of each of the different amino acids. But you can see that some are more common and others are less common than that uniformly distributed value of 5%. And in fact, you see things like the non-polar side chains, leucine, valine, alanine. Those are pretty common, and they're basically there to fill space and become the interior of compact globular proteins. Others are not so common. Histidine, for example, it's used fairly sparingly by nature. It turns out to be very important in the enzyme active site, and so it doesn't show up just anywhere. It's used very carefully by nature. And so what we can do in terms of trying to take this complex distribution and break it down into something that is a little easier to understand, we could divide this complex distribution of 20 amino acids into just two categories: polar and nonpolar, or in other words, polar and hydrophobic. And what's the recipe for life in terms of if we were to put all of these 20 amino acids into a category of either polar or nonpolar? It turns out that there's a very careful balance that's almost 1-to-1, almost 50-50. There's about 55% nonpolar or hydrophobic amino acids and 45% that are polar, or in other words, hydrophilic amino acids. Almost 1-to-1. In other words, in making globular proteins, what we're basically doing is trying to put in the interior the buried hydrophobic amino acids that will make a dense, compact core surrounded by the water-loving amino acids that are trying to be in contact with the aqueous solvent. So since we have excluded water from the interior of that globular compact protein, we have the possibility of having strong hydrogen bonds. Water would normally be a hydrogen bonding donor and acceptor, but if water is excluded, then the hydrogen bonds are going to be between the different amino acid residues of a protein chain. Well, here's a protein that's pretty typical of the globular proteins. It's lysozyme. And the structure is shown here, and I'm basically just slicing through the different colors of the amino acids. Are white is hydrophobic, red is the polar hydrophilic side chains. And you can see, to a rough degree at least, that the surface of the protein is hydrophilic and the interior is the compact hydrophobic amino acids. And so that's really the take-home message. The densely packed interior core is based on these hydrophobic amino acids, and in other words, we basically have an amphiphilic molecule. An amphiphilic molecule is a molecule that has both polar and nonpolar residues, and it divides itself in a way that will allow the polar residues to be in contact with water and the nonpolar residues to be buried. And the bottom line is that's the most important feature that determines the structure that drives the globular compact proteins to fold into these spherical-like shapes. Just for point of reference, since we're used to seeing them, I have drawn for you a rendering of the lysozyme in the typical way that you're used to seeing it, where you see that the secondary structural elements are shown there. The chain, the polymer chain, is shown as a continuous strand, and these two are exactly the same structure, the lysozyme structure, but in the upper right hand corner it's drawn out, it's shown, it's rendered as a space filling model, and here it's the typical kind of cartoon model that shows secondary structure.